Earlier, we talked about, from using the heat map view, identifying a region of interest, capturing that data from the heat map view, selecting a particular protein and wanting to look at the information for that gene. So I'm going to reiterate some of the things that you can do from the feature page. This is a breadcrumb across the top that describes the genome, and then below it describes the gene itself. Gene or protein in Patrick are sort of synonymous terms. This is the identifier in Patrick for this particular gene. It's 358, so it's saying it's the 358th gene on this particular genome. At RefSeq, this is what the gene is known as. Here's the gene symbol for it. This is what we call it in Patrick. This is the protein identifier. It's a hyperlink to the resource where you can get that. This is the gene identifier. This is what local family it belongs to. Here's the global family. Then we get into information about the genome, the taxon ID for Brucella microti. This is the unique identifier for the genome, which is that number 0.3, and this is what it's called. This is the accession number. Brucella actually have 2 chromosomes. This is the chromosome that this gene is on, with its start and end location and its orientation on the strand. So it's going in the forward direction. And generally, this is the way you describe a location of a gene on a genome. The nucleotide length, you could get the sequence here, the amino acid length, and the sequence here. We have some different tools that you can go outside of Patrick to look at. This isn't a very exciting gene, so this won't be very interesting, but in other genes you might want to do that. The local family will tell you how many genomes within the genus Brucella have this protein. The global family, when you click on this, it would open a new tab, will tell you how many genomes in all of Patrick have this. The Fig fam, that's a functional family, and it'll tell you how many genomes have that. You can see how many genomes have an identical protein sequence and how many have an identical nucleotide sequence. So by clicking on these, like I'll click on this one, I didn't do that in the last time, and it'll just show you how many have the identical sequence to it. And it may take a while to load. So not many. Not many of the Patrick genomes have an identical nucleotide sequence. Whereas if you look at the protein sequence, you'll see that 99 of the genomes have an identical protein sequence, or the genes in Patrick, but only 14 have the same nucleotides, meaning that there are different SNP-like values for that. So what I wanted to talk to you about today is using the Genome Browser. Earlier we talked about, here's the gene. It's always in red to draw your eye to it, and here's its gene neighborhood. And we talked about the Compare Region Viewer. But remember when we were at the heat map view and we captured this region, and these are the genes in this region, and this is the guy we liked. This is a region that's a genomic island in Brucella. And so you may want to capture the sequence for not just a gene, but for this region. How would you do that in Patrick? Well, you would need to go to this tab, which is the Genome Browser. So let's click on this. And it's highlighting in yellow the region of the browser view that corresponds to the gene you came into, which is this gene. Now let me point out that this is one of the few places in Patrick where you can do direct head-to-head comparisons of the annotations. Every genome in Patrick is annotated using the RAS-T-K pipeline. The RefSeq annotation, it could be Prokia, or it could be some other annotation pipeline. And each of those have different gene callers. They might have different rules. Annotation pipelines say every gene under 100 base pairs, we throw it out. They have different rules for when genes overlap, how much of an overlap you have between genes. So there's a lot of under the hood differences between annotation pipelines. But this is one place that you can see the RefSeq annotation and the Patrick annotation. And if you scroll over the RefSeq, you can see what the gene is called and what it's called in Patrick. This is called a hypothetical protein, as that is. So it's a kind of cool thing where you can actually see that. So let's go back here. And this one started with gene 349 and it ended with gene 365. So I want to go to the Genome Browser. Oh, well, this is 360 and 354. Somehow I need to expand this to capture what I wanted the task we're set upon is capturing the sequence that corresponds to this genomic island. So how do I do that? Well, here, this is a cool little thing you can do. See this little plus icon here? Let's click on that. And we can include in this table a number of things, including the start and the end for that particular gene. And then we need to unclick it. So for this one, 349, which is the start of that genomic island, and I click on that because I can't cut and paste anything from this table, but once it's highlighted, I can cut and paste from information beyond the green bar. So let's click, mouse over this and copy it, this 344965. And then we go here into the Genome Browser, and I highlight over that and I paste it in. There, okay? And let's see where it ends. We have to unclick it. And this is the last one, this 365. And then here is the end sequence. So I copy that and I go here and I paste it. And now we've got the region of the genome that corresponds here to the genomic island, and let's click go. You can see now we're getting, there's the 365 and there's the 349. Now it's not, this is JBrowse is a little particular in this way in that it always is a few off of nucleotides, but that's okay, we're close enough here. So how do I download the sequence? The next thing we're going to be doing in Patrick is blasting this data. Let's go here, and where it says reference sequence, let's click on that, and then say save track data. And then it gives you some options. You can save the highlighted region, which would just be that gene. You could save the whole sequence, which is 1.22 megabases. No, we don't want that. Let's do the visible region, okay? And you can either view it or save it. But let's look at it, view it here and see what it looks like. And this is the whole, and you can still save it at this point. And so that is how you would find the sequence that corresponds to that particular genomic island in Patrick. In the next section of this series, we'll talk about blasting that data. And thank you for watching, and thank you for using Patrick. Bye.